Okay, hello everyone. All right, so we're going to continue on our discussion on lipid bilayers, but we're really going to focus on the fluidity of the lipid bilayer. If we take a look at biological membranes, their basic structural element is basically the lipid bilayer. It forms spontaneously, and if we consider the structure of glycerols, sphingolipids, and sterols, they're amphiphilic molecules containing both hydrophobic and hydrophilic groups, but they tend to be insoluble in water overall. So what drives the formation of the lipid bilayer is something we talked about, earlier in the term known as the hydrophobic effect. If you put something like a glycerophospholipids in water, you have a number of water molecules that now will form this ordered shell around glycerophospholipids, where you basically decrease the entropy of water, which is not favorable. So what drives the formation of the lipid bilayer is the hydrophobic effect. The clustering of the different hydrophobic groups minimizes the number of water molecules that will otherwise form an ordered shell around the different molecules, all right? Which now then increases the entropy of the system if you have less ordered water molecules, which is a driving force for the formation of the lipid bilayer. It's also a driving force for the formation of micelles. All right. So overall, the lipid bilayer is more of a fluid-like structure where it's in constant lateral motion, right? If you have a suspension of glycerophospholipids and sphingolipids, they could form what is known as a self-sealing solvent vesicles, all right? Where within the interior of that particular vesicles, what you have is an aqueous environment. You have an aqueous environment right here, and the polar head groups are basically exposed to that aqueous environment. So because of this fluid-like property of the lipid bilayers, you can have lipid molecules or phospholipids can actually move across the lipid bilayer. They could move from one side of the lipid bilayer to the other side of the lipid bilayer, something known as flip-flop or transverse diffusion, which tend to be very slow, but they could also move across the lipid bilayer, and it's just showing you this orange head group, and its associated lipid is moving from one position to another, and that is known as lateral diffusion, which tend to be quite rapid. We'll see later that it's limited by a number of different proteins that are embedded within the lipid bilayer. It could also be controlled by sterile molecules such as cholesterols that are embedded within the lipid bilayer. Okay, so if we take a look at an example of a lipid bilayer, we have a purple color right here. Ta-da-da. We have the purple or magenta color showing us choline. We have the green that's showing us the phosphate head group. The gray is really showing us the fatty acid chain, which ends in a carbon, and the carbon atoms are shown in yellow. So the lipid bilayer overall is fluid-like, but we could also have sterile molecules that are embedded within the lipid bilayer, such as cholesterol. So that black one is an example of cholesterol in this case, all right, which, as you can imagine, would decrease the overall fluidity of the lipid bilayer and gives it more of a rigid-like structure. It will basically interfere with its motion. And that's just, think about it, they're in constant motion. Okay, so what might control the fluidity of the overall lipid bilayer. It's the fatty acid, the length of the fatty acid chain, the degree of saturation of the fatty acid chain. If you take a look at the table where you have the fatty acid chain, saturated, unsaturated, as the fatty acid chain increases, so does the melting point. So that affects the overall fluidity of the lipid bilayer. And it's affected by something we refer to as the transition temperature. Above the transition temperature, the overall lipid bilayer is what we call a liquid crystal. It's a crystal because it has some sort of order, and it's liquid because all these fatty acid chains are also in constant motion, and it can undergo flip-flop and lateral diffusion. But above that transition temperature, it ends up forming more of a gel-like structure. So it ends up being a lot more ordered. Okay.